All right, well, let's go ahead and get started. So I want to welcome all of you to the third webinar in our series being hosted by the PA Botany Committee. Thank you for joining us today. I am Maura McGinty-Kloss, and I am the Programs and Outreach Manager for the Natural Areas Association. And NAA is working in partnership with PA Botany to bring you this program and our symposium in November. And I'm delighted that you're joining us today for Small and Surrounded: How Population Size and Land Use Can Influence Patterns of Reproduction in Native Plant Across Remnant Serpentine Barren Grasslands of Pennsylvania. So for those of you who are new, who is PA Botany? So PA Botany is an education effort that involves botanists and plant enthusiasts from Pennsylvania, the surrounding region, who come together to inform and expand the knowledge of PA native plants and are committed to the protection of the region's native flora. The program was established in 2012, and PA Botany has hosted a convening of both amateur and professional botanists biennially in the form of the PA Botany Symposium, and then annually in the form of field workshops. This has been done in order to advance knowledge, appreciation, and conservation of our region's native plant heritage, and it's been successful thanks to the contributions of so many fellow botanists who offer their time and their knowledge. And it's just a great way to keep the study of botany alive in Pennsylvania. So our planning committee is a dedicated group of passionate volunteers who bring together these programs, and we're truly committed to making sure that 2021, in spite of COVID, Delta variant, all those things, we weren't without programming. So that is why we've offered the webinar series, and we'll be offering a virtual conference in November. So on behalf of everyone who's enjoyed past symposium or workshops, I just want you to join me in thanking our committee. So I mentioned our webinars. So we had two previous webinars. If you weren't able to join us, they're on the screen. Implications of Climate Change for Invasive Species by Dr. Bethany Bradley. The Design and Establishment of Seed Mixes for Prairie Restoration by Dr. Rebecca Barrick. And then, of course, today we'll be hearing from Dr. Spigler. So these are on demand. They're available at the pabotany.org website, and they are free for you to access and enjoy at your convenience. So we want to encourage everyone to register today for the virtual symposium on November 6th. We wish we could be together, but as we were planning this year, we just were uncertain, and it turns out it was probably a good thing. So we're going to do a one-day virtual symposium on November 6th. It's a Saturday from 1 to 4, sorry about that, 1 to 4. And there is no required registration fee. It's a pay what you can or pay what you would like to. So don't let that be a reason you don't participate. The agenda will include a keynote, cool finds, and a poster competition. And our keynote is Dr. Peter Zale, who is Associate Director of Plant Breeding and Collections at the Longwood Garden. And his presentation is Native Orchid Conservation and Efforts at Longwood Gardens. So we'd also like to encourage, it's been a few years. If you have some cool finds, some plants that you have cited that are unusual or an invasive that you want to report, that sort of thing, we are collecting those now. Go to the pabotany.org website and please submit some pictures. We're taking everything from 2018, 19, and 20. So please let us know what your cool finds are. And if you are in the academic community or a high school teacher, we are having a student poster competition, and we are still encouraging students to submit their abstracts or their brief descriptions, and we would enjoy having your students be a part of it. So we also would like for you to mark your calendar for 2022. We plan to be in person. Hopefully we will be November 4 and 5, 2022. So now I'd just like to take a couple of minutes. As I mentioned, the Natural Areas Association is working in partnership with PA Botany, and I'd like to tell you a little bit more about us. We are a national nonprofit membership organization dedicated to the support and advancement of natural areas professionals who protect biodiversity through science-based conservation and stewardship and advocacy. We're basically a community of professional land managers and ecologists and botanists and all those fields who work together to identify challenges, develop solutions, and share critical findings, and essentially advance the practice of protecting native flora throughout North America. So I also want to encourage you to register for the 2021 Natural Areas Conference. This will also be virtual. It's Tuesday, October 19th. It's 11 a.m. to 5 p.m. We're doing it in partnership with the Xerces Society for the Invertebrate Conservation. And we will be talking about natural and prescribed fire and its impact on insects and other invertebrates. So we hope you'll join us. You can find registration information at naturalareas.org. And then we're also hosting in November a webinar using informed plant selection to restore pollinators and songbirds in human-dominated landscapes. It's being presented by Dr. Desiree Narango, who's a wildlife ecologist and postdoctoral researcher from the University of Massachusetts Amherst. So that's on November 16th, and that is at the normal noon hour. So now we'll get to the reason for our gathering today. I want to introduce our speaker. So Dr. Rachel Spigler received her BS in Ecology and Evolutionary Biology from the University of Arizona and PhD in Botany from the University of Georgia. She is currently an assistant professor of biology at Temple University, where her lab focuses on the evolutionary ecology of plant reproduction. Since moving to Temple, she has conducted her research in the serpentine barren grasslands in southeastern Pennsylvania, with the overarching goal of understanding how land loss and land use influence plant reproduction, connectivity of plant populations among the remaining grasslands, and selection on floral traits, and ultimately population persistence. So with that, Rachel, I'm going to turn it over to you.
Okay, thank you so much. And despite practicing this, I did something to my computer. So hopefully you can see my screen.
We see it.
Okay, of course I can't see anybody else. So thank you so much for that introduction and for the invitation to be here. I'm really excited to be a part of this series and get a chance to speak to other plant enthusiasts about some of my research, understanding the consequences of human activities on plant reproduction. And it's funny, as I was listening to your introduction, which was very kind, I noticed I must have given you some of those details a bit ago, because I'm very happy to say that I am now an associate professor. So that's very exciting news. And so what I want to do today is tell you a bit about my research that is related to understanding some of the conservation implications of what's going on in southeastern Pennsylvania. Let's see. And in particular, a lot of what I'm studying is applicable really globally, because it's true that globally in this age of the Anthropocene, the natural world is facing multiple challenges. And perhaps the biggest challenge and the biggest cause right now of biodiversity loss is the sheer loss of habitats due to human activities. And these range from logging to agriculture, mining, pollution, and of course, humans needing somewhere to live, and urbanization of areas and urban sprawl. And this has made habitats not only become smaller, but scattered into disconnected fragments. Now, agriculture is responsible for about 80% of global deforestation, but even still, they represent lands that have some ecosystem services, and of course feed us, but even agricultural lands, which can still contribute to those services, are in decline due to urbanization. And so in the United States, for example, in just a 15-year time period, just an estimate from the American Farmland Trust, 4 million acres of land have been converted to high intensity development. And that means a high percentage of impervious surface, and another 7 million converted to low density residential development, kind of like what you see here with these very huge yards and houses spread very far apart. And the problem is it turns out that even these low density developments take away habitat that could have otherwise been useful, but they're also 23 times more likely to become urbanized compared to other agricultural uses. And in fact, Pennsylvania ranks 12th in the US among states where agricultural land is threatened. And so much of my research is centered around understanding the impacts of habitat loss and habitat fragmentation, as well as land conversion, and their impacts on plant-pollinator interactions in the remaining natural areas. And in particular, the ecological and evolutionary consequences for plant reproduction. So this is a figure I really like because it shows some of the interactions that occur due to both habitat loss and habitat fragmentation. There are both direct and indirect negative effects of these. And when we talk about habitat loss, I'm talking about the destruction of the natural system by humans, whereas fragmentation would be the breaking it into smaller pieces, that subdivision, where they can often become isolated. And we know that when we have habitat loss and fragmentation, that this leads to a decrease in plant population sizes, as well as of the pollinators ourselves. And so, let's see, am I clicking? So when we focus on these, we see that, what that means is that we're going to have plants perhaps suffering reproductive losses due to the fact that there's simply not enough pollen donors around. They simply don't have enough mates, and they don't have enough pollinators. And this is compounded by the fact that small populations are often less attractive to pollinators as well. So they get even lower visitation rates by pollinators, leading to an increasing breakdown of plant-pollinator interactions. And these interactions are critical, given that the overwhelming majority of flowering plants either rely completely on pollinators or to a large degree to enhance seed production in some way. And this includes agricultural crops as well. And so pollination represents an indispensable ecosystem services. And, it said you can thank a pollinator for 1 in every 3 bites of food that you eat. And of course, on top of that, I'm sure many of you are familiar with global declines of certain pollinator species due to human activities. And so when we combine this reliance on pollinators and the impacts of habitat loss and fragmentation, we expect and indeed find in many cases that seed production is diminished in wild plant populations. And this can lead to local extinction within habitat fragments. Now, fragmentation can also alter pollinator movements between habitat patches, as well as interact with population size to influence the movement of pollinators among populations and within populations. So one of these consequences, for example, is that in my own research, I've found that there's a strong relationship between population size and the probability of a plant getting the wrong species pollen due to the fact that they're relatively rare and pollinators are more likely to move among species. So this is the species I study, Sebacia angularis, and when plants are in small populations, they have a really high probability of getting that wrong pollen, which can also diminish seed production. And then finally, casting a shadow over these processes is the impact of land use in the areas surrounding remaining habitat fragments. And so landscape alteration, we're talking about the reduction of floral resources and nesting resources due to this conversion to agriculture, low intensity development or high intensity development. And it can also influence pollinator composition. So in my research, I've been studying these processes and dynamics in the serpentine grassland communities of southeastern Pennsylvania. And these are incredibly beautiful habitats. I'm showing you just a couple pictures from some of the sites around southeastern Pennsylvania. And when you think of the eastern United States, serpentine barren grasslands is not necessarily the first thing that comes to mind. And it's true, they are a very rare habitat on the East Coast, and they're rare globally as well. But on the East Coast, their center of their distribution, over 90% of the serpentine areas are in southeastern Pennsylvania going into Maryland. So this is a map showing that distribution in the mid-Atlantic United States. And serpentine grasslands, what they are is they are grasslands that are underlain by a particular type of bedrock, serpentine or ultramafic, that when it is exposed and weathers, leads to a soil that can be very dry, very high in heavy metals, and also low in many nutrients. And so this makes it inhospitable to trees. and also is toxic to many plant species. And so it becomes a reservoir for many forbs and grasses that can persist in these areas, and are also home to several rare plants and animals, including butterflies and moths in particular. So this is the distribution of the sites. And what I want to point out is that if you look at the squares, these are the approximate areas of the serpentine barrens. And I should state that the barrens can include serpentine woodlands, some of which have a high density of different types of pines, and the grasslands make a pretty small percentage of them. And you can see that some of these are quite small, and even many of them have become extirpated through activities like mining, urban development, and the disruption of natural disturbances. In particular, fire is something that has historically maintained these grasslands to be so, because otherwise you have the buildup of organic materials that then allow the invasion of larger shrubs and trees. So the existing, or I should say the remnant serpentine grasslands have become even smaller. And again, this is due to that forest encroachment that I just mentioned. Here are some aerial views of some of the serpentine sites. You can see the grasslands here by their outlined in this green color. So some of them are just a big swath within a development. Some of them have become mostly forest, and you can see them really scattered into these tiny little grassland patches. And this is a figure from Roger Latham and McGeehan, where they're plotting the grassland, the serpentine grassland area in a particular site, Unionville Barrens, from 1940 to 2010. And you can see how much of the grassland's been lost due to this, the interruption of the disturbance regime and the forest encroachment essentially kind of choking in these grasslands and making them smaller and even more isolated. And then finally, when we look at where these sites are nestled, so you have these grasslands nestled within serpentine forest, but then the forests themselves are in a matrix of human land uses ranging from agriculture to high intensity development, the development represented by gray here, and cropland represented by this yellow orange. And then you can see kind of some grassland areas peppered in between them. And so these serpentine barren areas make kind of an archipelago across southeastern Pennsylvania, and they vary quite a bit among different clusters that are surrounded by quite a lot more urbanization versus quite a lot more rural and agricultural development. Now, within these sites, I focus on studying a particular plant species that I've been studying for a while, and I think serves as a really nice model for understanding the impacts of fragmentation and land use on plant populations, because it's a generalist species. It grows on serpentine sites, but it's also found off serpentine, so it's facultative. It is visited by generalist pollinators, so it doesn't have a really tight specialized relationship. It's not dependent on any single pollinator. And they're also capable of self-fertilization. And so this is important because it also has implications for understanding what land loss and land use can do, not only to seed production, but how often inbreeding occurs, and in particular self-fertilization. It has a biennial life cycle, so I kind of am representing this here through these pictures. Any life cycle, we can start wherever we want. I'll start here with the flowers. So they tend to flower July, August. So many people don't see them because August is a pretty hot month to be hiking about in these open hot grasslands, but they create these just really beautiful displays of this kind of Barbie pink flowers that I mentioned are visited by generalists. They're nectarless. You find bees, mostly bees of different sizes, leafcutter bees, sweat bees, and syrphid flies are probably the most common pollinators. Occasionally you see maybe some skippers poking about, but they'll be pretty disappointed because this species doesn't provide any nectar. The fruits are dry dehiscent capsules, and then they produce these ridiculously tiny seeds, as you can see, that are almost like sprinkling salt on something. And every time I actually grow them in the greenhouse, I always think, there's no way this is going to become a plant. And life is pretty magical. And lo and behold, you get these adorable tiny little seedlings that overwinter. and then produce the flowers again. So it's an obligate biennium. It takes 2 years. And this is kind of pretty beautiful habitat where it thrives in this grassland, and you can see them like little pink lollipops standing out. And you can, as I mentioned, find it not just in serpentine areas, but in any kind of open area throughout kind of the eastern to Midwest of the United States. And although it's considered secure in many states, it is critically imperiled or in a few states in particular, because most populations are extremely small. They thrive, or I don't know if they thrive, but they survive in roadsides. And the only place you'll find these really large populations are in prairies where they're actively maintained. So there's some kind of mowing or fire, something done to maintain that open area. So I'm going to be telling you about a series of studies looking at understanding how plant reproductive patterns are altered across this kind of landscape where these serpentine barrens are situated. And I'm going to start by telling you about some pollination outcomes, specifically talking about seed production, but also talking about how the degree of self-fertilization can change due to a shift in these pollinator dynamics or plant-pollinator interactions, and even how selection can change. The evolutionary pressures can change among these populations due to these plant-pollinator interactions and their disruptions. And then finally, I'll tell you at a broader scale what's going on in the landscape and how movement between populations of genes of these plants, presumably by the pollinators rather than seed, can be disrupted and lead to increasing isolation of the remnant populations. So as I mentioned, habitat loss, fragmentation, the impacts that they have on population size and pollinator density should decrease pollination success. And indeed, we do see that in many species, but it turns out there's a big difference in which species are experiencing that problem. So most flowering plants are hermaphroditic, so they have stigmas that receive the pollen and have the pollen fertilizes the ovules. And of course, they have the anthers, which have the pollen as well. About half of these are capable of self-pollinating, either by themselves or with the help of pollinators. And the other half are self-incompatible. And so what this figure here is summarizing many studies that have been done on both self-compatible and self-incompatible plant species across different types of habitats that have been fragmented. And if we, and what these authors have done, it's a meta-analysis. And so they've summarized the average effect that fragmentation has had on the pollination success or the seed output of these types of plants. And they found that on average, we see a quite negative effect of fragmentation on seed production for these self-incompatible plants, but only a slightly negative to really overlapping neutral effects for self-compatible species. So this suggests that self-compatible species may be less likely to suffer from these disruptions in plant-pollinator interactions. And one of the proposed mechanisms for why this occurs relates to plants that are able to self on their own without the help of pollinators. So this hypothesis is that self-compatible plants may suffer fewer negative consequences on seed production, essentially because when no pollinators are around, they have the ability to do it themselves. And so this reliance on selfing in the absence of pollinators is called reproductive assurance. And the idea is that this would alleviate pollen limitation. And so this makes sense, but there have been relatively few experimental or experiments showing how this could relate in a fragmented system. And so that's one of the goals that I wanted to do with my work, is to understand if this might be occurring in Sebaceous angularis. What do we see happening to seed production, and are plants relying on that ability to self-pollinate? So in order to study these pollination dynamics, I did some floral manipulation studies that allow us to make inferences about how pollen-limited populations are and how much they rely on selfing. These illustrations show what the manipulations are. So I either had flowers in over 22 populations where we left flowers intact. And these are flowers that have both access to pollinators as well as their anthers, and therefore, if they can self-pollinate, they have the ability to do so. Then we also took flowers and we allowed them access to pollinators and access to their own pollen, but we supplemented them with outcross pollen from another plant within the population. And the idea here is that this flower should have the highest seed production because it's getting all the pollen it could possibly need from the pollinators itself and from us. And then we also took flowers that we emasculated, meaning that we kind of snipped off the anthers. And this is important because this represents a treatment where the flowers still can get outcross pollen brought by pollinators, but they no longer have the ability to self-pollinate. So this allows us to quantify different pollination parameters. And the first of these is called pollen limitation. And essentially what pollen limitation is quantifying or addressing is it's saying, well, how much is seed production reduced because flowers aren't getting enough pollen? So we are comparing these pollen supplemented flowers that have all the pollen they could need to seed production from flowers that get it from pollinators and themselves potentially. Then we can also quantify reproductive assurance, which is a measure that is quantifying how much seed production is reduced when we eliminate a plant's ability to self-pollinate. So in other words, if plants are depending on themselves, as well as pollinators, that when we take away that ability to depend on themselves by taking away their anthers, if we see reductions in seed, then it must mean that these plants are selfing in order to provide reproductive assurance, in order to boost their seed production. And so we can do these manipulations in the field. This is, these are buds that we've emasculated, and we can indicate the different treatments by painting the pedicels different colors. These are some fabulous students I had work in my lab on this project and doing it, and you can see it in action. And then when the fruits develop, we can come back and collect it. The paint colors stay quite nicely, so we can match them back to their treatment and look at seed production. So when we did this across 2 years, we did in fact see evidence of pollen limitation, meaning that plants were not getting enough pollen. If they had more pollen from pollinators or additional mates in the population, they would do better. But that's not consistent from year to year. So here's pollen limitation, again, comparing these pollen supplemented flowers to control flowers. And we see that in the white bars, which is representing the average across years, plants are pollen limited. But this is primarily driven by higher pollen limitation in 2014 and low to essentially significantly no pollen limitation. And similarly, we also see that plants are relying on that ability to pollinate themselves when we look at this quantification of reproductive assurance. So again, on average, we see significant reproductive assurance, but this is driven what's going on in 2014 as compared to what's going on in 2015. So we do see these dynamics shift from year to year. Now, my main interest was understanding about how this plays out across the landscape, because we have populations from those figures I showed you where these serpentine grasslands are nestled in areas that are highly developed to primarily forested. And they also range in population size from just a couple of individuals to thousands of individuals, depending on their patch size and how they're managed. So I wanted to look and see, well, to what extent does the amount that seed production is pollen limited, or the degree to which plants rely on reproductive assurance, how much are they influenced or driven by variation in population size? And how much is it driven by the differences in how much land is developed within a radius? I chose a 500-meter radius. So this is just showing you the extremes. A patch of grassland you can barely see here that is within a county park. And a grassland right here that's in a kind of gated community with a shopping center right here that's pretty highly developed, nestled in Westchester, Pennsylvania. And so what we found was that certain populations are depending on reproductive assurance much more so than others. And in particular, it's those small populations that are highly developed, right in this corner here, that are relying most heavily, up to 30% increase in seed production when they can rely on themselves versus when you take that away, compared to populations that aren't developed, for example. So you can see that interaction here. And that what seems to be going on is it's this ability to self-pollinate that ultimately evens the playing field among populations with respect to the amount of pollen limitation they're having. So you still see a pattern where these small populations surrounded by high levels of development do seem to experience a little bit more pollen limitation, but by and large, it's highly depressed by the fact that they can self. And so this is reassuring in the sense that these plants have a way to combat pollen limitation, but it does change how much they're selfing versus outcrossing, and I'll touch on that later on. Now, when we consider mechanistically, why is this happening? What's going on in these small populations surrounded by high development? We looked at a few potential causes. One of course is, well, maybe they're not getting visited enough. And so we looked at pollinator visitation rates, and very briefly, I'll say that we did find differences, but not necessarily what you'd expect. We actually found that pollinator visitation rates were higher in small populations, and they actually decline in large populations. And so this might seem surprising at first, but if we imagine that perhaps pollinator abundance, when we group all the pollinators together, is the same or not very different across these different patches, what I think is going on is essentially you're seeing competition for pollinators among very large populations, because there's only so many pollinators to go around when you have thousands of plants, whereas there's plenty of pollinators perhaps to go around in these small populations. But that doesn't mean you have plenty of mates. So that can also contribute to pollen limitation despite having high visitation rates. We didn't see a significant trend with developed land, although it is noticeable that you don't see any high visitation rates in these highly developed areas. So instead, what I think is going on is that it's related more to the composition of pollinators that we're seeing in these different patches, and in particular, where we see higher rates of development. So as I mentioned, this plant is visited by a variety of pollinators, mainly bees, so bumblebees, small carpenter bees, belly bees, which are among my favorite, or the leaf cutter bees and sweat bees, as well as syrphid flies. And we also get others, including this little beetle right here, which seems to actually be a bit of a pollen consumer. And so presumably bees are the better pollinators, and we have some studies that I'm not showing you the results of to suggest that. And when we look at how the proportion of visits made by bees varies, we see that it does decline significantly when you have more developed land. So some of the dynamics that we found seem to be driven by this change in pollinator composition when you have higher development combined with the lack of mates in small populations. So I then wanted to ask, well, okay, these are what's going on ecologically, but what do we know about, or what are the implications for evolutionary pressures that these different populations might be experiencing? And could that mean that there is selection for different traits in these small, highly developed populations where plant-pollinator interactions aren't as strong compared to large grasslands that are primarily surrounded by natural habitats? And so studying this is relatively straightforward, although extremely labor intensive. And so one of the first things I needed to do was get some measure of plant-pollinator interaction strength across the populations that I've studied. And I did this in a subset of populations. And so how we did this was we used pollen deposition as our metric. So across these populations, I went and I collected these plant stigmas, and we were able to count. You can see this imaging software, all these blue dots represent pollen grains on the slide and on the stigma, and we can quantify how much pollen these plants are getting. And presumably plant populations with stronger plant-pollinator interactions are the ones that have higher pollen deposition rates, and those with weaker plant-pollinator interactions are going to have less pollen grains. We also looked at a suite of traits that we were interested in related to pollinator attraction in particular, with the thinking that, well, perhaps where these plant-pollinator interaction strengths are low, that there should be greater selection for traits that improve a plant's ability to attract a pollinator. So we looked at traits like floral display, how many flowers might be open at a given time, how large the flowers are, how tall they are. Some of them, they're usually, oh, I don't know, maybe 30 centimeters, but they can get really tall, almost a meter. And then I also wanted to consider whether or not or when plants were flowering. If we think about each of these being a flowering curve of a given individual, right? It starts flowering, it opens most of its flowers, and then it declines. You can see that, we have this pattern across the population where the majority of plants are flowering together, and we would say those are synchronously flowering. And then you have these ones that are kind of off the beaten path and are asynchronously flowering, we would say. They're either flowering earlier than most or later than most. And one of the ways that we can get a measure of the strength of selection is to measure fitness of these plants. And we can ask, does their fitness, fruit production or seed production, does it change if you change the trait value? So for example, do plants with larger flowers, do they make greater seeds, more seeds? Or plants that have larger floral displays or flower synchronously? And the slope of this line, importantly, tells us how strong selection is. The steeper the line, it tells us that selection is stronger for this trait or for this population than it might be for this population. Okay, so these are the results from that study, and I'm going to walk you through these figures. They're another set of three-dimensional figures. And I'll just warn you that the way that they're set up, this is kind of a reverse axis where you have lower values on the right-hand side of the particular trait of interest. This example here is petal area, going up to larger flowers towards the left. On the y-axis here, I guess it's the z-axis, I'm showing you fruit set or seed set. We measured fitness both ways. And then on this axis here is our measure of plant-pollinator interaction strength. So here, low values would mean weak plant-pollinator interactions. High values we would interpret as strong plant-pollinator interactions. And so this figure here shows you the results from petal area. And how we interpret this is we can see a steep line or a steep decline in fitness as flowers get smaller. And we see this for these populations that have weak plant-pollinator interactions. And it's hard to see, but if we flip it around, okay, and we're now looking at the backside, we see that this ultimately levels out such that there's weak to no selection for larger flowers in populations that have strong plant-pollinator interactions. So in other words, where plant-pollinator interactions are weak, we do see strong evidence for plants making larger flowers doing better than plants making smaller flowers. But in these populations with strong plant-pollinator interactions, it doesn't matter what flower size you have, all the plants are doing fairly well. We see a very similar pattern when we look at plant height. And now for plant height, it's not a floral trait, but if you are growing in grasslands and you are, it can be a measure of apparency to pollinators because short plants will be pretty much covered by the surrounding grasses, whereas taller plants will nicely pop out above. And again, we see the same pattern. We also found really interesting patterns for these two traits, flowering duration, how many days a plant is flowering for, and that synchrony measure, are they flowering in sync with everyone or out of sync with everyone? And what we actually found was it wasn't just the strength of selection that was changing, it was actually the direction. So whereas in populations with weak plant-pollinator interaction strength, we saw selection favoring plants that flowered for longer periods of time and that tended to flower together, so more synchronously. But that completely shifts when we look at populations we consider to have strong plant-pollinator interactions. So you can kind of see that in the back here, where now selection seems to be favoring plants that flower for shorter periods of time and that flower asynchronously. And so some hypotheses I have about what's going on here is if we think about what's more important to plants, well, if plant-pollinator interactions are weak, then there's going to be stronger pressure to get the attraction of those pollinators or to get more pollen. And so flowering synchronously, being larger, flowering for longer periods of time, that is what's going to increase your pollen receipt. On the other hand, if you're already in a population that gets plenty of pollen, all right, and you have strong interactions with your pollinators, then getting pollen is not the problem. Instead, something else begins to be the problem. And in particular, well, there could be many things, but some things that could be going on is that plants that flower for longer tend to dip in later in the season where resources and water start to become limiting. And so if you have enough pollen, don't kind of drag it out, flower for short periods of time and avoid that. Or it could be to avoid competition for pollinators, as I mentioned, that can happen in some of these large populations, or even to avoid predispersal seed predators, which I see in these populations. So that kind of tells us a bit about what's going on within each of these patches in relation to how much seed they produce and what kind of selection pressures they're facing. But I also wanted to take a broader look at the landscape and understand what's going on to movement between populations. So in other words, gene movement across the landscape when we consider this backdrop of what the remnants are nestled in. And for this particular species, and for many particular plant species, pollen can be, is really the main way that genes are passed from population to population on the bodies of pollinators. And this is particularly true for a species like Sebaceous angularis, where if you recall the size of those seeds that I showed you, they're extremely tiny and they're gravity dispersed. So they're not going very far. And so the majority of gene movement across populations should be through pollen. So we can think about what might promote the movement of pollen between populations versus what might retard it. And it has a lot to do with this matrix. And so this figure here, I think, nicely illustrates what's going on when you have populations of different sizes and different patch sizes that are occurring in either a matrix that's favorable to the movement of pollinators or that's not favorable to the movement of pollinators. So in this example, we have our cute little bee here. That's our main pollinator that will pollinate the flowers in this population, but will also disperse to other populations. And in doing so, deliver pollen. And this would occur for small populations, perhaps medium size and large. But once you are in a matrix that's inhospitable to pollinator movement, then pollinators are more likely to stay within that patch. And that may be fine for this population, but for smaller populations that might critically rely on the influx of genetic material to maintain genetic diversity and the ability to adapt, if they're cut off, they can often experience decreases in genetic diversity, higher levels of inbreeding, and a reduced adaptive capacity. So it's important to understand how pollinators are moving across the landscape as much as what they're doing within each patch. So I used genetic markers in this study where we did it's called a landscape genetic study to try and understand that movement by looking at the genetic connectivity of these populations. And the basic idea is that if there are no barriers to movement for pollinators across these populations, across the matrix, then we would expect that the only thing determining how distant these populations are genetically would just be space, right? The farther apart they are, it's just less likely that they're going to exchange genes because the movement of pollinators is not infinite, it is limited at some point. And so we can use genetic markers to estimate genetic distance or gene flow, and we would expect that gene flow is going to be lower between any two populations the farther they're apart. And we can measure that gene flow with a measure called genetic distance, right? So the farther the populations are, the more genetically distant they should be. This is just determined by space if the matrix is hospitable. But if it's not, and it's a more hostile matrix, then it shouldn't just be determined by space, but rather what is in between these fragments that we're looking at. And could it be, agriculture versus an urban landscape, and to what extent are they resisting it? So we can measure resistance to movement. And so I think about how, if you think about resistance to our own movement, right? And you picture what it might be like the resistance to running on a treadmill versus then going to run on a road or running through mud, or then apparently deep water running is a thing. You can find anything on the internet. So if we kind of look at how this might play on the landscape, we can measure, how long, you can think about how long it would take someone to run from one patch to another. And it's going to take much longer if they have to run through larger patches of water or larger patches of mud than if they just had a long treadmill getting them from one place to the other. So in an analogous way, right, that's how bees or how pollinators might be seeing the landscape. And in particular in these grasslands where these bees are nesting, where they're foraging, if you have a large grassland, it should be it shouldn't provide much resistance to their movement and thus the pollen that they're carrying. As you get through woods, this is still suitable nesting sites, and there are some floral resources. But then as you increase or you encounter agriculture to low density housing to very high urbanization, we can imagine that this is kind of going to be like that running through mud, where we're going to see less movement of pollen between populations and therefore greater isolation. So we can quantify this and we can ask, well, is the matrix, can we quantify these different types of land use and ask, is that what is determining how much gene flow is occurring between these populations? So in this case, we would say that if we have this resistance presented by different land uses, then however we quantify that cost, that's going to be what's determining how related or unrelated populations are and how much they exchange genes. So again, this is just to remind you the matrix in which the serpentine grasslands are nestled. There are two regions, the Westchester Barrens that I study and the State Line Serpentine Barrens. And again, this is the highly urbanized area where you have, Westchester and some agriculture and some open spaces. And then you go towards Lancaster where it's much more agricultural. And this just shows you an aerial view again of these two extremes of what it looks like, though it's not to scale. So the kind of the glitch in doing these types of studies is, okay, well, I can make analogies like running through mud or sand or water, but we don't actually know what the cost of traversing a low density development versus a high density development is for a pollinator or a pollen gene or a seed. And so in order to do these studies, we need to make some hypotheses about how much it costs to move across different landscapes. So for example, we can assign different costs and we can use, and then we can test these with the data that we collect. So in all of the cases, we're assuming that it doesn't cost anything for a pollinator to move through grasslands or shrublands. And the cost of whether it can move through the forest, well, it depends what kind of pollinator we're talking about. Maybe it's the same as moving through a grassland, or maybe these are pollinators that tend to deflect when they get towards any kind of forested edge. And then with cropland, again, it's going to depend what type of cropland we're talking about, the distance of the pollinators. Maybe it's twice as hard to move through cropland as it is through forest or shrubland, or 4 times as hard, or 100 times as hard. And similarly, we can do the same for low development and high development, right? Is it the same, no impediment, or is it extremely difficult for a pollinator to move between it? Does it resist movement 100 times that of grassland and shrubland? And so if we look at the connection between these populations, we can quantify this cost distance depending on how much of these types of land uses are intervening, and try out different costs and see what the data seem to match. So when we did this, we found that there is indeed a cost to different land use types, but they're very different depending on what type of land we're talking about. So if we're talking about forested land, we never found, or none of our data supported a model that suggests that these natural forests are an impediment to connecting these populations for gene flow. But we do see that cropland seems to be twice as hard for genes to cross than forest or grassland. And consistently, that low development is twice as much as cropland and 4 times as much as forest. And I'll just say that when this table, what it's showing you is what we call our top models, our models that explain the most amount of the variation. And in fact, by incorporating land use and not just including distance, we were able to explain 3/4 of the variation in the genetic distance we saw across populations. So an increase of 25 percentage points over what distance could do alone. What this means is simply that these models perform equally well. So no matter what model we looked at, forest, cropland, and low intensity development always had these values suggesting that they were the best supported by the data. High development, it was a little, different models were equally good, but no matter how you slice it, what we found was that high development is anywhere from 10 times to 200 times more difficult for gene flow to occur across it than forest or grassland. And so when we look at a graph of our top model of what this might look like, we see a really strong relationship between kind of what's going on, how the land is resisting movement across it, compared to genetic distance, with populations having greater amounts of intervening development, having much higher genetic distance than otherwise. And then as a consequence of this, what we found is that these highly isolated populations, the ones that are surrounded by greater development, have lower genetic diversity, and they have higher values of inbreeding. So we are seeing these consequences of being small and being isolated, not only in the ecological consequences of the reduction in seed production and these changes in self-fertilization, but also in their genetic diversity. So to summarize, what I've found in this work in these in southeastern Pennsylvania and these really amazing habitats is that the surrounding land use has a huge impact on its pollination dynamics as well as population size, and that these actually interact. And that even though it seems that, or I've found that these plants can compensate the lowered seed production by selfing, this itself can have a demographic consequence. I only looked at seed production, but some of the work I'm doing now is following these plants throughout their entire life history, because I also know that when these plants self, those selfed plants are have very low fitness compared to seeds produced through outcrossing. So that's work that I'm doing now. And then finally, my work also has shown that at least in this area, the populations seem to be able to withstand a little bit of cropland or low intensity development, but that high intensity development is just completely cuts them off. And that it seems that corridors can even be helpful for the movement of not just animals, but the genes of plants that they might carry. And I'd just like to thank the PA Botany Symposium Committee for the invitation, and co-authors, many students, many landowners who have just been wonderful in letting me access these habitats, and of course, my funding. So thank you very much.
Well, thank you, Rachel. That was really interesting. And you packed in a ton of information in a very short time.
I hope not too much.
No, it was really interesting. So we do have a few questions. So we have about 6 minutes. So let's run through as many as we can. First question, how can you be sure that some traits like larger petals aren't just attributable to inherent site habitat conditions like soil fertility, sun, shade, et cetera?
So, I'll answer that in a couple ways. So the variation, we find very similar variation in flower size across these populations. So it is true that maybe at a particular site, if it's not, homogeneous and there's a plant on a patch of high resources and therefore it has larger flowers. I mean, it may be plastic, but the pattern of selection that we see just tells us what's being favored. If it's plastic and not genetics, then maybe the population won't respond to that selection, but we do see this favoring. Now, it could be that they're favored and they have higher reproduction because bigger flowers make larger seeds. And so I have a series of studies that I've done that I'm working on now, where we've done some extra experiments to see, is it really the pollinators or is it resources or something else? But the fact that we see this relationship between how strong selection is and how much pollen these plants are getting on average is pretty consistent with it being, pollination dynamics that are strongly influencing selection.
Okay. What are inbreeding cost effects when selfing is utilized to counter pollen limitation?
Right, so I extremely briefly showed that, and I know that in this species, self seeds may be 80% less likely, or I guess I should say, from seed, A self seed is 80% less likely to survive and reproduce and produce seeds itself than an outcross seed. But I also know that that varies across populations. So in some populations, it's 80%, in others it's 20 or 30%. And so that's one of the things that I want to understand is what is the cost, not just for selection, should plants self more, should they not, but what are the demographic costs? If you're already small and you're surrounded and isolated and you're selfing more, are you just gonna is population growth just gonna tank when you add this selfing? And that's something I'm investigating.
Okay. Does this species have any predispersal seed predators whose activities might counteract selection on flowering time, plant height, those sorts of factors?
Yeah, absolutely. So there is, I have one in my freezer. I need to find someone who can identify it, but we were very excited. We found it and decided to rear it, and it became a moth. So it's some kind of moth. The larvae, one larvae per capsule, just eats all the seeds and eats a hole and makes its way out. And I do suspect that's what might be driving selection for asynchrony or shorter flowering duration in these populations that are getting plenty of pollen. I would like to test that, but I do think it plays a role that I suspect is quite interesting.
Okay. When you assess stigmas, do you find other species pollen or level of pollinator consistency?
Yeah, so in small population, what I've found is that in small populations, plants, like 60% of the plants will have at least some heterospecific wrong species pollen on them. And in large populations, hardly any do. So there's clearly an impact on, some of it might just be sheer relative abundance of Sabbatia versus other species. And it also could be that pollinators will move differently depending on the density of plants. But I see a strong impact of population size on whether they get the right pollen or the wrong pollen.
Okay. Just a note that PADCNR is able to use our C2P2 grant program to fund habitat restoration projects. And they recently closed out a serpentine barrens to use the C2P2 grant program to fund habitat restoration projects. And we're curious to know more about the state of native pollinator habitat in natural areas where there's a lot of competition. Would management include nesting sites for native pollinator be beneficial to these sites? So,
well, I know someone, if he's still here, who would be much better qualified to answer that than me if Jeff Allerton's still here. But, I know from the plant side, certainly this management, right? This keeping it open, keeping the invasive trees and shrubs out and leaving it open just makes a huge difference on the plant populations. For example, I see this in Sabbatia. Once they burn it, the next year they'll have 1,000 plants kind of pop up. There's a seed bank, which makes it more complicated. But yeah, there needs to be a maintenance of pollinator habitat that is integrated into how they're maintaining these serpentine grasslands.
So we have time for one more question, and it seems like a good one. Since Sabbatia angularis is found in habitats beyond serpentine barrens, would your findings apply more generally to the species in other settings?
Yes, I can actually say that. So I've also I started working with this plant when I was doing my PhD at University of Georgia, and I studied it throughout Georgia, North Carolina, and South Carolina. Now, as it turns out, one of the North Carolina sites was this kind of disjunct patch of serpentine, but the rest were not serpentine. They were different kinds of prairies. And we still see this, we see a negative impact of reduced population size on seed production and increased selfing rates.
Okay. Well, thank you so much. This was a wonderful presentation, highly informative. I know that many of our viewers are going to be coming back and watching it again just to be sure that they catch everything. So thank you for joining us today, and thank you to everyone who joined Rachel and me for this presentation. And don't forget to register for the symposium on November 6th with PA Botany. And then also, if you've got a cool find, submit it. We want to know what's cool. We want to know what you found. If you've probably had a lot of time outdoors during COVID, share with us what you've seen. So thank you all. And this recording will be posted within 24 hours at least, if not sooner. So thank you all very much.